Exendin-4 promotes Schwann cell proliferation and migration via activating the Jak-STAT pathway after peripheral nerve injury by María Isabel Osorio Bastidas and Pedro Vallejo.
The peripheral nervous system is a network of 43 pairs of motor and sensory nerves. These nerves control the functions of sensation and motor coordination. So in peripheral nerve injury, there is a common clinical disease. It causes the partial loss of segmental function of these nerves. So in this trial, they wanted to know more about the methods used for the PNI repair, but they wanted to know more specifically about the mechanisms of Exendin-4. Knowing that Exendin-4 is a good therapeutic agent because it can promote the regeneration of the nerves in the PNI.
Methods. The first of the methods was experimental animals. 68 8-week-old mice were chosen and divided into 3 groups. The first 18 were used for functional testing of the sciatic functional index. The other 48 were used for glucose measurements in blood, monitoring and micro-range assessments. Second, PNI model. The 48 mouse were divided into two groups. The first crush group, which was administered 0.5 milliliters of saline per mouse per day, and the other crush group was made of the other half, was administered 0.5 microliters of Exendin-4 with 0.5 milliliters of saline per mouse per day. These were monitored for 7 days, and after the 7 days, the sciatic nerve was retested and tested.
Method number 3, they got functional assessment of SFI. SFI is sciatic functional index. With this, they wanted to confirm the role of Exendin-4 in repairing mouse with sciatic nerve injury. So they divided 18 mice in 3 groups, the first one with Exendin-4, the second one without Exendin-4, and the third one it was a sham group. With this, daily they calculated their SFI, and the measurements they got were the distance between the heel to the first toe, the distance between the first to the second toe, and the distance between the second to the fourth. In the method number 4, we got the blood sugar monitoring. So glucose levels were measured continuously for 7 days, and the measurements they obtained were the glucose level at the 0 hour before the crush, blood glucose level fasting, and 2 hour postprandial blood glucose level after the crush.
Micro-experiment. RDNA was extracted from the sciatic nerve, it was purified and then turned into a double-stranded cDNA, which was hybridized to using the microarray assessments. A difference in gene expression between the two groups was defined with a fold change of higher than 2.0 and a p-value lower than 0.05. This was visually displayed in volcano plots, heat maps.
In the method number 7, what they used, it was a polymerase change reaction. This technique is very useful when you need to amplify a DNA fragment, because after that, it's much easier to know how the disease is behaving. In the method number 8 of cell culture and identification, they wanted to confirm the role of Exendin-4 in Schwann cells. So they divide 3 groups of the Schwann cells. The first one, it was a placebo group. The second one, it had Exendin-4, and the third one, it was a sham group.
Cell proliferation. Equal number of cells of the 3 groups were incubated at 50 micromolar of EDU for 2 hours. After the incubation, using a fluorescence microscope, 5 random images were selected of non-overlapping regions. This was used for calculating the DAPI positive cells and the EDU positive cells proportion. Cell migration. Cell migration was used for testing.
The method number 11, it is the Western blot analysis. They used this technique because they needed to identify specific proteins in a complex mix of proteins. So they did this after 3 steps. First, separating the proteins by size. Second, transfer to a solid support. And third, the visualization by labeling the proteins with specific antibodies. In the method number 12, for the statistic analysis, the data from 2 samples and from multiple set of samples were compared by using the student's t-test and the one-way analysis of prior.
7 of the 8 mRNAs were eligible for analysis. In Figure 2A and Figure 2B, all the samples were submitted to a PCA. which 4 of the samples from the experimental group and 3 of the samples of the control group were grouped in the same cluster. In figure 2D, the differentially expressed genes were visualized using a scatter plot, and in figure 2E using a volcano plot. A heat map shows that the 108 mRNAs with significant difference in expression were detected. 146 of those DEGs were upregulated and 34 of them were downregulated in Schwann cells injected with Exendin-4 compared to control Schwann cells.
Western blotting to detect the key proteins in all the process, and also we can see that the levels of Jak-2, STAT2 and STAT3 were not different between the groups after 24 hours. And in the other hand, the levels of PJAK2, PSTAT2 and PSTAT3 were higher in the group with Exendin-4. This can tell that Exendin-4 can activate the PJAK and the AG490 can block the activity of Exendin-4 by inhibiting the Jak-STAT pathway.
Schwann cells were cultured and identified by staining with 800. In figure 6C, we can see that the proliferative capacities of Schwann cells in the Exendin-4 only group were significantly higher than those of the Schwann cells in the control group. At the same time, the Schwann cells cultured in the Exendin-4 with the Jak-STAT inhibitor showed the same proliferative capacity as the control group.
The transwell experiment showed that the migration was higher in the group with Exendin-4 than in the other group without it, and also it showed that the difference between the group with Exendin-4 and AG490 and the control group wasn't that high.
Discussion. We took some of the authors mentioned in the text and their statements and said if we agree or disagreed according to the results of the article. Perry and Jolivet said, Exendin-4 is a good candidate for therapeutic agent that promotes nerve regeneration in peripheral nerve injury because it can attenuate peripheral sensory neuropathy, at which we agreed. Sarah Hahn said, after suffering the impact of peripheral nerve injury, Schwann cells transform their cellular gene expression pattern to create a microenvironmental variable for instrument generation of the axon, in which we agreed. Clemens and Du said Jak-STAT signaling plays an important role in the biological process of Schwann cells, which play an important role of the recovery of the peripheral nerve damage, to which we also agreed.
Conclusion is that thanks to molecular biology and molecular experiments, we can tell that Exendin-4 it regenerates nerve after PNI because it activates the Jak-STAT pathway. Also, we can tell that molecular biology has propelled medicine to a new level because it let us search from a new point of views, a lot of point of views, everything about the disease and everything that we have to know so we can treat it. Also, we can tell that molecular biology has been really helpful for everything that it has to do with physiological mechanisms in our body. So it's a really useful tool for us now in medicine.